It's my great pleasure to introduce Isabelle. So Isabelle is the postdoc in our team, and she leads the genetics analysis of our cannabis and me study. And it is really my great pleasure to have Isabelle for the first time in my own town and at this conference. And Isabelle usually has the very hard task to explain genetics to our MSc students, and she can do that with them. I'm sure she will succeed spectacularly well with us as well. So Isabelle, this is for you.
Hi, thank you very much for the introduction and for having me. I'm Isabelle Austin-Zimmerman. I'm a postdoc at the SGDP at King's College. And as Marta said, I'm going to be hopefully explaining or talking a bit about psychiatric genetics. So there have already been a few mentions of genetics throughout the conference. We heard about genetic correlation and Mendelian randomization on day one. We've heard in Robin's talk yesterday a bit of a trailer for this talk, looking at the GWAS and polygenic risk scores for schizophrenia. So hopefully today will give you a bit more of the background and context for what all of those words mean and why they're relevant for psychiatric genetics. So just a quick history. So people have been studying, the concept of things being inheritable since really the classical era, I guess. But the sort of start of what we think of as modern molecular genetics is really with Mendel's work on plant hybridization in the 1800s, which probably most people will remember from biology lessons at school. And here's, why is it going? It's giving away all the, here we go. Okay. So yeah, he discovered or made some discoveries about Mendelian inheritance, or what we now call Mendelian inheritance. And it was a bit ignored for a while, but around the start of the 20th century, we had developments in understanding of chromosomes and this idea of genes being the unit of inheritance. But the actual molecular nature of what are genes wasn't known until a bit later. When in 1953 we had the discovery of the double helix structure of DNA, which probably everyone has heard of. You've got Crick and Watson here, and obviously had to highlight the valuable or essential contribution of Rosalind Franklin, who was working at King's College London, and her X-ray of the DNA, which is really what provided the conclusive proof that Crick and Watson's theory was correct. And then the next kind of major moment that I'll mention in the history of human genetic study is the sequencing of the human genome, which was a long project. I think originally it was meant to be just 5 years, but ended up taking a lot longer than that. And just around the start of the 21st century, we saw the successful sequencing of the human genome. So assuming that most people have at least a basic understanding of what we mean when we talk about the genome, but it's basically 3 billion nucleotide base pairs long. So those are those A, C, T's, and G's, the letters that are organized across 23 chromosome pairs, which you probably all know a bit about. But what's really crucial to understand is that of that 3 billion or so base pairs, only about 1% or so is coding that actually writes the instructions for the genes, which write the instructions to make proteins. The rest of it, some of it is, regulating gene expression, and quite a lot of it, actually, we don't really know what it's doing. Or probably some people have more idea than I do, but a lot of it is unknown. And importantly, about less than 1% of those base pairs, of those 3 billion base pairs, actually vary between any two people. The amount that one person varies from the reference genomes is changeable, but it can be as little as 0.5% or maybe even less. But yeah, a very small amount actually is different between humans. But that's the part that we're interested in, because that's the part of our genome that's going to give us some insight to why some people have different experiences to others, why some people experience diseases and others don't. So I'm going to talk about 3 types, broadly, of genetic studies today, some which have been more successful than others. Firstly, talking about family studies. So these are sort of natural experiments that take advantage of either monozygotic or dizygotic twins that have at least some or all shared genetics and a shared environment often, or adoption studies where you have some shared genetics but no shared environment. And by studying these different groups, you can get a sense of what is the contribution of genetics to a trait versus the environment. The first example of a twin study was by Francis Galton in the 1960s, which is one of his more positive contributions to the field. I obviously am not going to be giving a history lesson on eugenics, but he was a renowned eugenicist. So otherwise, apart from some positive things, we don't like him. And then just to say, I won't really be talking very much about family studies today because it's not the field that I work in, but this type of study still plays a really important role in genetics and in psychiatric genetics. And I've just included one example here of the TED study, the Twin Early Development Study, which is based at the SGDP at King's, founded by Robert Plomin and now run by Talia Ely. And this is still providing some really valuable contributions to our understanding of psychiatric genetics. They're doing some genome sequencing as well, and they've also launched the CoTED study, which is looking now at so this has followed twins from born in the early 90s, and now they've got the Children of TED study, which my son has contributed some DNA to, who you might have heard in the back, who's now been removed for misbehavior. But yeah, a really valuable study that's still ongoing. The next type of study that some people might have heard of is talking about candidate gene studies. And this is an example of a less successful early idea about how to study genetics. The idea seems to make good sense, where you take what you know or suspect about a disease or trait and look for genes that are relevant to that, and see if there are mutations within that gene that might explain why some people experience the disease and others don't. The first successful example of this in terms of psychiatric genetics was in the early 2000s with a link between depression and the 5-HT transporter. And this same group with our very own Robin Murray also sort of showed an association between the COMP gene and psychosis. But even though I think we saw that this paper, one of Robin's most highly cited papers with over 1,800 or so, was it, citations, that he now, and it's wrong, yeah. So, I mean, I don't think you said even a review quite recently included it as a true finding, but generally, these are now accepted as largely they haven't been replicated at all, or very few examples have been replicated, so we now don't really believe any of these findings. But this is what sort of set the scene for genome-wide association studies, so taking a hypothesis-free approach, looking across the whole genome to try and understand a bit more about disease, traits and diseases, and that's what I'll be mostly talking about today. But before I explain, what is a GWAS and why we use them, I think it's important to have some context to the type of genetics that we're studying here. Often when we think about what is a genetic disease, people will often find that they're more familiar with what we would think of as rare mutations and rare diseases. So a lot of people will have heard of the Huntington's gene, for example, and this is an example of a rare gene with complete penetrance, which means if you have the risk mutation in that gene, you will develop Huntington's. I think everyone who has that develops the trait. There's other examples like the BRCA1 gene, which has got a lot of attention in recent years, that is also rare and has high but incomplete penetrance. So I think about 80% of people with this mutation will develop the trait. In psychiatry, we're interested in common variation. So these are positions on the genome that vary very frequently between individuals. And individually, those variations on their own have a very small effect size. But taken together, they can contribute to an overall significant increase or decrease in your risk of a disease. And what we would describe all psychiatric traits, and indeed most biological traits, as complex traits, meaning they are explained by a combination of many genetic markers and, importantly, environmental factors as well. So this is a nice sort of diagram of the steps involved in conducting a successful GWAS. And of course, the first step, as in any study, is in data collection. And in GWAS studies, this is no mean feat because we really need very, very large sample sizes. So in the realms of tens, if not hundreds of thousands, if not probably millions, eventually is what we're going to really need to be able to discover everything that there is to discover about these traits. So once you've collected your hundreds of thousands of samples, the next step is to genotype their data. So take a blood sample and do the genotyping. This is a specific type of study of DNA where, as opposed to genome sequencing, where you take an individual sample and you look at every single position across the genome. When we do genotyping, we're just looking at an actually very, very small proportion of our whole genome that we know commonly varies between individuals, sometimes even just 500,000 single nucleotide polymorphisms, or SNPs. So these are the variants, those letters, A, C, T's, and G's that can vary between people. So you look at a small sample of those, and then you do various quality control steps, one of which involves, as this graph shows, trying to group people into some fairly sort of heavy-handed, large, inaccurate groups based on where people are from in the world. And I'll hopefully explain a bit why that's relevant, but that step is essential for doing the next step, which is imputation. So I said when we genotype, we're just looking where those sort of darker dots are at certain positions on the genome. When we do the imputation, we try and fill in the gaps. And we can do that because we know quite a lot about the genome now, or at least about this part of the genome that varies between us. And all of those SNPs, A, C, T's, and G's, they're not inherited independently from each other. They're inherited in blocks. And we know quite a lot about those blocks. So if we look at one or two positions within a block, we can quite reliably fill in the gaps based on what we see there. We can say, well, if there's an A there, there's likely to be a C, T, and G next to it, and so on. Those blocks, LD blocks, linkage disequilibrium blocks, we'll just think of them as genome blocks. The length of those and the frequency of the variants within them can change a lot depending on where people are from in the world, which is why this previous step, wherever the little thing is necessary. So with a European genome, for example, those blocks tend to be a lot longer and a lot more similar to each other. And with an African genome, the most diverse genomes, those blocks are much, much smaller. So that's a necessary step. Then once you've done your imputation and quality control, you're ready to actually do the association test. And we obviously have heard a lot about GWAS already, but really what a GWAS is you can think of it as doing millions of regression association tests over and over again. So you're asking how much does this individual locus on the genome contribute to the risk of a trait. And you ask that question, 8 million times or so, however many SNPs we tend to look at, around 8 to 10 million usually. And then you look at the output. And once you've got the output of that, you have an effect size for each position, a standard error p-value, exactly as you'd get in any regression test, but you've got millions of them. Then you might be ready to do some meta-analyses. As I said, sample size is really important for these studies, so meta-analyses are an important tool. Hopefully you might be able to do some replication, and then you're ready to actually look at the data, interpret it, and do various post-US analyses, some of which I'll talk about today. We've seen some examples yesterday from Robin of a Manhattan plot, but this is really the main kind of primary figure that a GWAS will have that summarizes all those results. And just to go over again what it's showing us, each dot is organized, represents a locus on the genome, and is organized in terms of physical position along the genome, so from chromosome 1 to 22. And then on the y-axis, you've got the inverse log of the p-value, which means that the most significantly associated loci, those with the smallest p-values, will go to the top, and you, hopefully, if it's a GWAS with lots of findings, you get these nice peaks that look like the Manhattan skyline. So that's a Manhattan plot. And just to highlight the point I was saying about large sample sizes, so each of these SNPs have on their own very tiny effect sizes. And we're doing millions and millions of tests, so anyone that's done any statistical analysis will know that we have to do a multiple testing correction because otherwise a p-value of 0.05 would be not reliable. So we set the threshold for genome-wide significance as 5 times 10 to the minus 8, which is a correction not for all 8 or 10 million tests that we're doing, but for roughly the number of blocks within the genome that I've mentioned that are independent. So we're trying to correct for the number of independent tests. So that's why we need the large sample sizes. So you can see here, this is an example of the first Psychiatric Genetics Consortium, PGC, depression GWAS in 2013, which had nearly 19,000 people, so still most other fields, a really significant sample. They found 15 genome-wide significant loci, which was still an improvement on previous efforts. But if you compare that to David Howard work, who led this most recent PGC depression paper, who's at the SGDP, a sample of over 800,000 people and over 102 genome-wide significant loci. And then in schizophrenia again, so I think this was the example that Robin mentioned yesterday, that when this was presented at the World Congress of Psychiatric Genetics, and there was 7 genome-wide significant loci, that was so exciting that people cheered. But you can see now with a sample of 320,000, and we have 287 genome-wide significant loci. So a big improvement. But you might have noticed that the depression GWAS had a much larger sample size than the schizophrenia one, more than double the sample size, but less than half of the findings, which is sort of hinting at the idea that not all of these traits are equally heritable. So the variance explained by the genetic markers that we're looking at is not the same for different traits, which of course is not surprising. So this is a graph of the SNP-based heritability estimates from many of the recent PGC papers on different psychiatric traits. And the SNP-based heritability is really just the variance explained by all of the SNPs that we include in the genome, in the GWAS. So you can see actually the smallest amount of variance explained is depression, and the largest, even though it's actually the smallest sample size, is OCD. And schizophrenia is pretty near the top with 24% heritability. So we can see from this that the depression and other traits are less heritable than schizophrenia. So we'll need even larger sample sizes, well into the millions probably, to discover everything that there is potentially to discover about the genetics of depression. And also still need larger sample sizes for schizophrenia as well. Some people might have heard that schizophrenia is around 60 to 80% heritable, and that is still true, even though I've just told you that it's 24% here. That's because the SNP-based heritability is sort of, we think, a subset of the overall heritability, and we essentially think that we haven't found all of the variants that explain the heritability for schizophrenia. So family-based studies have estimated the contribution of genetics to be between 60 and 80%. And last year, actually, was the first time that a GWAS study found all of the missing heritability of a complex trait. So there was a GWAS on height, where they estimated the SNP-based heritability to be 50%, which matches what had been previously estimated from family studies. And for that, they needed a sample of 5.4 million, which is, of course, you know, a metric like height is included in basically every study, so that, you know, any clinical study or data collection or biobank. So that's just giving us some indication of how much work is still to come in this field. But that was obviously very encouraging, because it kind of confirmed that indeed, really, this is essentially a power issue, and that there is much more to be discovered. And then just wanted to show this figure, which people might have seen an example of before. There's been a few different iterations of it. This is from the most recent study looking at this last year. And here is showing, this graph is showing the genetic ancestral background of participants or samples within GWAS studies, compared to how that compares to the global population. And you can see, probably it won't surprise anyone, that by far the biggest chunk of people in GWAS studies to date are European. which massively does not correlate to the proportion of people, the proportion of the global population that is European. This obviously hinders the generalizability of any of our findings, because we know that we treat people from a range of backgrounds, so most of the findings from GWAS are really only reliable for Europeans, if they're reliable at all. And apart from this being an ethical issue, this is really hindering our ability to discover, the truth about the genetic component of these diseases. Those blocks I mentioned briefly earlier, the blocks of inheritance that are inherited vary in length between people depending on where they're from, with the most variability and the smallest blocks being within Africa. The reason that that's important in terms of discovery is that when we do these association tests and we're looking, if we find a SNP, a variant within a block that's associated with a trait, say schizophrenia, and that block is very, very long, we actually don't really know which variant within the block is causing the disease, or which handful of variants perhaps within the block are causing the disease. We can only say that there is one here that is causing it, and we mostly pick the one with the smallest p-value, but that doesn't necessarily mean that it's the causal variant. And you can imagine that if you've got the very, very long European blocks, you're further away from knowing which of those is the causal one, versus a much smaller block in the African genomes and a scale in between is going to be easier to try and find. And crucially, what's important is studying diverse groups of people, because if you're seeing the same variants coming up across all of your population groups, we're much more confident that that's a truly causal variant. So just wanted to highlight that that's a really significant problem within the whole field of population genetics. So that is hopefully some background to give some understanding about what is GWAS, and now to try and put that into the context about why we would care about that. So how can we use that data to potentially learn more about diseases? So there are lots of ways that we can try and use the data to do that, but one of the first things that you might do is you might try and take those summary statistics from your GWAS, those individual SNPs, and say, okay, well, that's just a collection of millions of SNPs. What do they really mean? What do they do? And map those to the genes that they are either in or near, so potentially have a role in regulating. This is an example of some of our work on psychosis, where we've done that, and this time it's another Manhattan plot, but this time each dot is representing a gene rather than a protein coding gene, rather than a SNP. And then using that, you can then look up what do we know about the function of these genes and the proteins that they code, and that can give you some indication about the disease and the causes of it. So, for example, some of these genes that are to do with calcium channels come up, genes with a role in neuronal transmission. Then you can, take those genes and think, and look at what biological pathways those genes then play a role in. And again, using our psychosis work, we've done this and shown that many of the significantly associated genes cluster and play a role in the pathways to do with developmental biology. I think these are quite small, the text, but developmental biology pathways, the neuronal system, and metabolism. So, for example, some of the genes are to do with neuronal development, to do with calcium channel signaling, and other pathways relevant to neuronal transmission and neurodevelopment. So just some examples of how we might use this data to learn more about what is actually causing the diseases. Then we're obviously interested in how can we use this data to help with prediction, identifying people who might be at greater risk of developing a disease or trait. So polygenic risk scores we've already heard a bit about. This is where you take all of those variants that we've found through a GWAS that are associated with a trait, and you then look in a different sample and calculate for each individual a risk score, which is a weighted sum of the effect of those associated variants. So this can be very valuable in terms of, you might take, for example, the relatively well-powered schizophrenia GWAS with 300,000, and most of us don't necessarily have access to our own samples of 300,000 people, so you can then leverage the power of that data and apply that to your potentially much smaller sample. So we have done that, for example, in the EU GAI study, and looked at the schizophrenia polygenic risk score and how this distinguishes cases from controls. And you can see that I made very small down here that the mean polygenic risk score for cases is significantly higher than for controls. But crucially, what you will also see on this graph is that there are cases with very, very low polygenic risk scores, and there are controls with very, very high polygenic risk scores. So this is not a diagnostic criteria. What we're doing really when we talk about polygenic risk scores is taking a population-based metric and applying it to an individual, and translating that to an individual absolute risk is not straightforward. And if you think about if you're told that your polygenic risk score for schizophrenia is in the 90th percentile, that might seem very alarming. But if you're then told that the polygenic risk score on its own explains maybe 5 or 6 to 9% of the variance between cases and controls, then that's quite a lot less concerning. It doesn't mean that you're 90% more likely to develop the trait than someone else. There has been there is ongoing a lot of work at the SGDP, partly led by Oliver Payne, to help think about how we could translate these scores to individual risk, because a lot of people do want to do that. You can understand the value that would have. But it's obviously very complex, and trying to quantify the impact of environmental risk factors is obviously challenging and going to vary from trait to trait. And crucially, a polygenic risk score is only as good as the data that builds it. So I've already explained the limitations that we have even from the very best GWAS within psychiatric genetics. We know that we haven't discovered everything, so there's an automatic ceiling on how much the polygenic risk score can explain. And very importantly, the polygenic risk scores for non-Europeans are far less predictive of case control status. This is a graph showing that where you've got relative to Europeans, you can see how predictive the polygenic risk scores are for other populations, and it's really lacking. So again, a very long way from being able to implement these clinically, but that is, presumably the eventual goal. There you go. Despite that, they still have value, they're still useful, especially for research. So this is also some of our work on the EUGI sample. And you can see that when we add polygenic risk score to the model in terms of predicting case control status, you do see a significant improvement in the predictive ability of the model, which is great, but also this graph helpfully highlights that really the biggest improvements come from adding additional environmental factors. So I think at the moment, the most valuable way to use these scores is to combine them with environmental risk factors to try and get an overall picture. Okay, so I'm sure most people know, whether you work clinically or not, you can imagine that it's not very common, I suppose, especially within psychiatry, for you to see a patient who has one extremely clearly defined single condition, and that's the only condition you worry about for their whole life. Most patients will have comorbidities, or maybe it's not even that easy to diagnose their specific condition. And we can use GWAS data to learn a bit more about the relationships between traits. So one of the ways that this is often done is a reasonably simple analysis where you look at the genetic correlation. So what is the overlap of the genetic risk between two traits? So this is some of my work on sleep genetics, and you can see that I've shown that both long and short sleep are associated with, have significantly positively correlated with PTSD, with depression, with cannabis use, bipolar, negatively correlated with bipolar, and also with physiological traits like coronary artery disease and BMI. So we are getting a picture that there's a shared genetic architecture for some of these traits. That's sort of a global metric, but what you can then do is look on a local level, so look in either specific regions of the genome or in specific genomic pathways and see is there a correlation between those traits there. And there's various different methods that will allow you to do that. And we have seen already an example of this figure that is a nice map of the genetic correlation between psychiatric traits. And you can see that this helps to kind of cluster based on what you might expect to see in terms of the symptoms of these traits, with a very high positive correlation between bipolar and schizophrenia. So 70% of the genetic architecture of these two traits is shared. Slightly higher with about a third when you look at depression, 50% shared genetic risk for anorexia and OCD, a negative correlation between ADHD and anorexia, and the thickness and darkness of the line is representing the degree of significance. So this is work from the PGC again. So yeah, helping this just showing how genetics can help us understand the relationship between diseases.
Just a quick technical question. Sorry, sorry to interrupt you.
It's okay.
Just a very quick technical question. Are these univariate and multivariate, are these partialized correlations? So, for example, the negative correlation between anorexia and ADHD, takes into account the rest of the correlations, or
no, I think these are pairwise comparisons.
These pairwise kind of univariate correlations. Okay, thank you very much. Sorry.
So we also heard a bit about Mendelian randomization. So this is which I won't explain too much behind, but it's a type of causal inference study that attempts to sort of mimic a randomized controlled trial, which we know would be the kind of gold standard for assigning causality. But you can use genetics to sort of mimic that and understand if there is a causal relationship between some of those traits where there's correlations. So taking, for example, again, some of my sleep work, looking at depression, where we've shown that there's a bidirectional causal association between long sleep and depression, but only going from short sleep to depression, not the other way around. And we have also looked at schizophrenia, and there's, again, a bidirectional relationship with long sleep, but this time only a relationship going from schizophrenia to short sleep. So giving a sort of indication that of the potentially causal relationship between these traits.
So, if there's a causal relationship between sleep and schizophrenia genetically, does that mean that you can do anything about it? I mean, if you have a genetic predisposition to short sleep, and that is related to schizophrenia, if that were true, if you actually slept long, deliberately, would that help you, or you can't do anything about it if it's genetically correlated?
Well, I think, again, the relevance is thinking about the variance explained by these genetic factors. So both sleep and schizophrenia, for example, are heritable traits, but again, there's multiple other factors that actually explain a far greater proportion of the likelihood in getting the diseases. So I guess this would be, the interpretation is that these traits have a shared genetic risk, and that being a genetically high risk of being a short sleeper can increase your chances can cause you to develop schizophrenia, but the variance explained is still pretty low for those traits.
But if you had one where the variance was quite big, can you do anything? I still haven't understood. Is the genetic correlation means that you're going to get a higher risk of schizophrenia, whether or not you actually try to sleep more?
I don't know if anyone has I guess, I don't know if you'd be able to, I think so, because I think for any of these traits, the heritability estimates for sleep are smaller than schizophrenia, for example. So I think, and that's even true, I guess, with family studies. But yeah, I don't know how people have, I guess you would need quite good longitudinal studies to follow that up, but yeah.
Sorry, there is another Murray who has something to say on the topic.
Oh, yeah.
Thanks. That was a very interesting point. Can you hear? Yeah. So I guess you'd need, I suppose the only way of really knowing might be to do a trial, which obviously would be very difficult to do, but that might help disentangle a genetic correlation from a causal pathway.
Yeah, I think
Thank you.
definitely that would be the ideal scenario. And I'm sure there are trials ongoing, but I don't know how it probably, well, I don't know, they did mention there are sleep deprivation studies in depression, aren't there? So,
well, and
I guess this would not really support that. But
yeah. And you were talking about short sleep towards schizophrenia. I think not sleeping long is in general bad. So I would recommend if you're not sleeping long enough, sleep longer, and then if you prevent schizophrenia, that's a good unexpected outcome.
Yeah, everyone have a lie-in. So obviously, really, what we're talking about, kind of everything I've talked about, is an example of where we care about gene-environment interactions. A lot of the time when you read a GWAS study, they really do just focus on the genetic component, but in all psychiatric traits, the environmental risk factors are significant, if not more significant than the genetics. So understanding the relationship between a genetic risk and an environmental risk and how those interact is really kind of what we all want to work towards. There's been many candidate gene by environmental risk factor studies which have the same pitfalls that we discussed earlier and haven't necessarily been well replicated. There are new methods, I think one came out in 2020, where they try and do a genome-wide by environmental risk factor association study. But you can imagine the challenges with that in terms of sample size. I think recently I heard someone say that you need 8 times the sample size for an interaction study to what you'd need for a main effect. And if you think of the case of depression, for example, we've already got a sample of 800,000 people, and we haven't even explained all of the variation, all of the genetic risk factors. So having a sample size large enough for, oh no. Oh, there we go. That was a quicker resolution. So the sample size burden is really large. The methods that exist obviously try and account for that, and you can do certain things by only take the genome-wide significant loci only and sort of partition it that way, but it's still quite a challenge, especially for psychiatric genetics. No progress?
Definitely Tony's fault sitting next to the apparatus.
Oh, here we go. Okay, so one way you can try and overcome some of these power limitations is to use a polygenic risk score again, where you're sort of aggregating that effect of the GWAS. And this is just one example of some of our work on schizophrenia and cannabis. We're looking at the interaction, and you can see each of these lines represents a different level of cannabis use, with the daily users of high-potency cannabis at the top, rare and never users at the bottom. And you can see that as your polygenic risk score increases, the risk of being a psychosis case also increases, regardless of in line with your cannabis use. So one example of how people can use genetic data to think about gene-environment interactions. Then, of course, we have epigenetics. So this is the study of heritable changes to gene expression, so not changing the A, C, T's, and G's, not changing the base pairs, but modifying the way that the genes are expressed through DNA methylation, histone modifications, and other methods, but those are the sort of 2 most common ones. And there are examples of where this has been clinically relevant. I think most epigenetic studies within psychiatry at the moment are still animal model studies, but I mean there are human studies, but I think the most conclusive findings are from animal studies. But everyone's probably familiar with the example of smoking, where you can see very significant differences in DNA methylation patterns, histone modification patterns between smokers and non-smokers, or with age acceleration. So this idea of biological age and the fact that your DNA methylation patterns can be predictive of your likelihood of getting disease and of overall mortality. So now to briefly talk about genome sequencing. So as I said at the start, that all of the data when we're doing GWAS studies is using genotype data, where we're not looking at every position on the genome, we're looking at a specific subset, a small subset, and filling in the gaps. And we've discussed some of the limitations of those methods. But GWAS can miss vital information. So as I said, 90% or thereabouts of the genome is not very well understood. Rare variants, because we're filling in those gaps, we're filling them in using statistical methods, it's not really possible always to distinguish between what is a real rare variant that someone has and what's a mistake or a statistical error. So we have to kind of discard that. If there's a variant that within your sample is seen in less than 1% of people, we typically just exclude it from all our analyses, which obviously is going to miss some information. Especially as some of those rare variants on their own could be the ones that have much larger effect size, as I explained, the difference between rare and common variation. And structural variants can be hard to capture with genotype data. So this is where you have regions of the genome that are replicated or deleted. That can be quite hard to measure with genotype data. People do, it is done, but it's not always so reliable. And I mean, the reason that we've used genotyping instead of genome sequencing, which obviously is substandard, is really the cost. I think only recently the cost of sequencing a genome came under £1,000. I think it is coming down quite rapidly now, but still, £1,000 per genome when you need samples into the millions is obviously not feasible for most funders. But these are it's missing potentially very clinically significant things. So those of you who work in schizophrenia might have heard of the 22q deletion, which gives rise to DeGeorge syndrome, which can increase your risk of developing schizophrenia by 25%, I think. And that's where a region of the genome on chromosome 22 is deleted. It's also very important for pharmacogenetics, which I'll come on to in a minute, where we have genes that we know are very relevant to drug metabolizing, and there are some significant structural variants within those genes that are in pretty much all clinical studies of these genes are not captured. And again, this has especially great significance for the study of rare variants and non-Europeans, because of course when we say a rare variant, generally what we mean is this is a variant that's rare in Europeans, and very often they're not at all rare in other populations, but we're still not looking at them. So there are programs now. There's the newborn genome program is doing whole genome sequencing of 100,000 babies, led by Genomics England. And then you have efforts like UK Biobank, which I think has already released the genome sequence for 200,000 people, and I think are releasing them for everyone, which is going to make it obviously an even more valuable resource than it already is. So then finally, to talk about pharmacogenetics. So I mentioned some pharmacogenetics just now. People might be familiar with the CYP genes, cytochrome P450 enzymes, which are liver enzymes, a family of liver enzymes that are responsible for metabolizing most drugs on the market, I think at least 75%. And you have these two genes, CYP2D6 and CYP2C19 especially, or enzymes, genes that encode the enzymes, that metabolize the vast majority of antidepressants, antipsychotics, and mood stabilizers. And these are highly polymorphic genes. So there are between any two people in this room, the variants within those genes are likely to be quite different. And some of those variants can impact the way that the resulting enzyme functions. So you have poor metabolizers whose enzymes of one or both or several of the CYP genes are working slower than usual, or slower than we'd think, and then on the other end of the spectrum, ultra-rapid metabolizers where they're working too fast. And you can imagine the clinical importance of this, where if you're metabolizing a drug much more slowly, that drug is lingering within your system for longer, you're going to be at greater risk of toxicity and of side effects. And on the other end of the spectrum, if you're an ultra-rapid metabolizer, that drug might be moving through your system before it has a chance to really have an effect, and a larger dose might be required to achieve the same effect. So some recent work in the UK Biobank came out which showed that basically everyone has one of these variants in a known pharmacogene. So either those two CYPs that I mentioned or some other known genes. And 24% of people within the UK Biobank had been prescribed a drug for which we would expect them to have an atypical response. So this is a really significant proportion of people, probably potentially could affect everyone in this room, basically. Now, in terms of studying these, this is some of my PhD work where we did a meta-analysis looking at the effect of these different metabolizer groups, whether you're a poor metabolizer, ultra-rapid, or in between, on potential side effects of antipsychotics. And even though these are very small, you can probably see that there's no effect in these meta-analyses looking at prolactin levels or weight gain. And that is partly probably explained by the fact that none of these studies, none of these primary studies were capturing the full extent of the genetic variation within these genes. So again, where genome sequencing or even targeted sequencing efforts where you specifically look, which is much cheaper, and you specifically look at these genes, is going to be very valuable. There are other ways that we can use genetic data to study treatment response. There have been examples of looking at the genetics of treatment resistance. So obviously a significant problem in both depression and schizophrenia, for example. There is some evidence from the heritability estimates from the schizophrenia study, for example, that perhaps those people who are treatment resistant, the heritability is higher, so the variance explained by genetics for those people might be greater. So the sort of goal of this type of study is to try and identify, are these representing distinct phenotypes? Are these practically different groups of people, or is it a more clinically severe illness, sort of on a spectrum? So far, the GWAS on these have been underpowered and have not found any genome-wide significant associations, but this is, as all psychiatric genetic GWAS, is an ongoing effort that will hopefully in the future lead to some important discoveries. Another example of looking at the genome-wide basis of treatment response, so again, work led by Ollie Payne at the SGDP, looking at antidepressant treatment response, and I think this is UK Biobank and maybe meta-analyzed with another sample, and showing at least that these are heritable traits in terms of remission or percentage improvement, and that these heritability estimates, again, are higher than for depression alone. And then looked at the variance explained by polygenic risk scores for treatment response, or looked at how various polygenic risk scores predicted treatment response, and actually found that I think the GWAS for schizophrenia and educational attainment predicted more strongly than the depression GWAS, which may be potentially a result of the differences in power of the GWAS that underlie those polygenic risk scores. And then finally looking at drug repurposing. So there was some recent work that showed that drugs with genetic support are twice as likely to be successful in clinical trials. So this was sort of retrospectively looking at drugs that had been approved that also you could identify a genetic support, so maybe through a reliable GWAS hit in a receptor for a drug, or in something to do with the transporter or within the metabolism. So this is obviously very promising to show, because this wasn't trials that used that genetic result, but if you look retrospectively at what's been more successful. So you can imagine the appeal to drug companies if you can say that you can double your chance of being successful in clinical trials if you have GWAS support. And as well as this, looking at drug repurposing, there are some efforts where you can look at drugs that, or GWAS hits that are relevant to a certain drug that might be used for another trait. Maybe anxiety, for example, if you look in the anxiety GWAS and you see that there's a significant association in a receptor for lithium, for example, this isn't true, but just as an example, then you might think, well, that's a really good candidate for repurposing the drug and testing the drug. So sort of providing a bit more support for some potential new drug development programs. And obviously, repurposing an existing licensed drug is cheaper than a normal, than a completely novel molecule. So yeah, just the GWAS, transcriptome-wide data, pathway analysis, Mendelian randomization, and polygenic scoring are all methods that can be used to hopefully improve drug development. And just to say that there are efforts to implement these, what we know about pharmacogenetics clinically. So this is the largest consortium, the CPIC, in the US, which drafts many guidelines for how to take into account pharmacogenetic information. And I think this is going to be increasingly relevant as more and more people actually have access to this data themselves through consumer genetic testing. So a lot of the currently widely available consumer genetic tests will tell you what your metabolizer status is, for example. So you might find in the future that people come to you with this data that they've got themselves. So having guidelines on how best to use that is obviously important. And this is being introduced in the NHS, including for some psychiatric drugs. But yeah, crucially, really the complexity of these gene-environment interactions really require large and prospectively designed studies, because as I mentioned with those meta-analyses that I showed, a lot of the studies of this are not capturing the full extent of the variation. So yeah, because a lot of the available data and biobank data maybe doesn't have, information on dosing or treatment duration that you would really want. So finally, in summary, just trying to think across the lifespan of a patient and how GWAS can be useful as they move through their life. So firstly, talking about risk prediction, so identifying those people that might be at greater risk. That could have potential use for prevention methods. So, if we identify someone's at greater risk of a disease, maybe we encourage them to avoid certain environmental risk factors. In terms of once we get to a diagnosis, understanding more about that diagnosis and what's caused it, the molecular basis of the disease, helping with treatment in terms of understanding who might be matching people to a better treatment. If they're a poor metabolizer, maybe you can pick them, give them a drug that is not metabolized through that enzyme. Understanding more about the potential genetic basis of treatment resistance, whether this is a distinct subgroup of patients, and understanding people's comorbidities and the shared genetic risk and biological pathways that might underpin those. So thank you very much. Thank you, my team, and our dogs.
Thank you very much, Isabelle. Question, comments? Rose.
Thank you very much for your talk. A while back, there was something about the HLA website, the HLA site linked to complement, and that might have a role in aberrant pruning. Is that still thought of as a possible thing, or is that fallen by the wayside?
I think the role of the HLA region is definitely still quite thought to have a role. I think you could have seen the significant peak on chromosome 6, which is where that would be. But there is a lot of complexity within that region of the genome, and it's actually excluded from a lot of post-GWAS analyses. So I actually am not sure where that particular theory stands. I don't know if Robin knows any more about it.
So this is to do with that huge peak on chromosome 6 in the Manhattan plot of schizophrenia. And a guy called Steve McCarroll had evidence that it was partly explained by the complement genes. And when it came out, this was the idea that this could be affecting synaptic pruning in a huge proportion of people with schizophrenia. But I think it's still holding up for quite a small proportion of people. So it might be a small proportion of schizophrenia might be related to synaptic pruning. But actually, I should defer to Graham. You don't believe it?
Oh.
No, he doesn't want to be, leave him alone, basically.
So it's basically, it's a very complicated region of the genome, the MHC region, and it actually is a lot of the post-US analyses, including the ones that I presented of our work, we remove that because it becomes very difficult to interpret. I would guess that genome sequencing data will help even more with that. But there's definitely GWAS support in that region of the genome.
Last question, unless people put their hand up quickly after this.
Thank you so much for your talk, especially since I feel like a total amateur when it comes to genetics. It seems to be so complicated for me. One thing piqued my interest at this moment. You said that treatment resistance may or may not be heritable. Might that also be for the opposite, so that treatment effectiveness might be heritable? So should we also, if we have a family of patients who suffer from, for example, depression in different generations, ask what substance helped one generation may also help the other better than others?
Yeah, definitely. Definitely that can be true. The work I showed by Oli Payne was looking at treatment response, which did show that that was heritable. I think that's probably one specific area, you know, if you have a specific family in mind, I guess, that where looking at those pharmacogenetic variants could be really valuable. You know, if you have a patient who you find to be a poor metabolizer at CYP2D6, and it's say depression, for example, and they haven't responded to any of the treatments, but everything you've tried them on was metabolized through CYP2D6, or maybe it makes more sense, the ultra-rapid metabolizer, they haven't responded. Or if you have patients who are suffering particularly more than you would expect from certain side effects, that could be an example of where to look. And I mean, you can do some of that within the NHS already, but I'm not sure what the criteria are, but that's what those guidelines that are going to be coming in are going to help, either whether, you know, if you have that data because the patient brings it to you, or guidelines on when it would be appropriate to test for it.
So,
hello, thanks for the interesting talk. You mentioned consumer data and these sort of studies like 123andMe, and you can purchase these. Is there any way for universities, for research to access the data, to buy the data? And or is it good enough quality?
Yeah, so 23andMe, for example, contributes quite significantly to a lot of these GWAS. I can't remember if they're in the schizophrenia one, but they're definitely in the depression one. Especially for traits like depression, which have a higher prevalence in the population, they do provide that data for research, assuming the participants consent to that, which makes it a really valuable resource. It is harder to access than other biobanks, and also commonly with GWAS, if you do a GWAS, you make available the summary statistics for that so that other people can take that and do further analyses. So the psychosis work that I presented, for example, was not using any data that we collected ourselves. That's using summary statistics from GWAS that have been run by the Pharmacogenetics Consortium, which represents a large meta-analyses. With 23andMe, they tend to limit the availability of the summary statistics, which is, I guess they have a legal reason for.
So you have to pay quite a bit of money.
Yeah, so and I'm sure you have to pay. That's.
So the question is, if you want to do your genome privately, what would be the best?
Well, I suppose if you, if outside of knowing someone who's running a genetic study like us, where you could maybe give us your data and some useful data on cannabis use, for example, which you're all welcome to do, especially if you're non-cannabis users. But outside of that, I suppose the best way is to contribute to already, do 23andMe, which is probably the best one at the moment for providing data for research, and make sure you tick that you're happy for your data to be used, and know that it's very difficult even for legitimate researchers to access, so the data should be very safe. But yeah, if people want to do that, I think 23andMe is the only big organization that I know of that provides that data. It can't be the only one, but it's the one that is definitely most widely used in research.
Yeah. So sometimes you see a paper and it says there is a highly significant genetic relationship between schizophrenia or depression and immune genes, or between the genes for schizophrenia and cannabis use. Mm-hmm. And the newspapers say the reason why people take cannabis is because they're predisposed to schizophrenia. But when you read through the paper, it says the polygenic risk score for schizophrenia explains 3% of the variance in cannabis use.
Yeah.
And I never know whether we should care about that, or whether, because I think sometimes I say, well, that's insignificant. But then people say, but the polygenic risk score only explains 20% of schizophrenia, so 3% is actually quite a lot. So,
yeah, I think obviously the word significant is used, can mean a few different things. It can mean actually clinically significant, and it can mean having a small p-value, and those two things are often correlated, but obviously not the same. And a lot of the things that I presented have very, very small p-values, but not measurable clinical effects, for example. I think, in our work looking at the EUGI sample, for example, having a high polygenic risk score for schizophrenia doesn't predict your likelihood to use cannabis or to use more heavily. But there is overlap of the genetic pathways that are involved in some of those traits, in those two traits. But yeah, again, like disaggregating the individual risk from this sort of polygenic risk score is obviously challenging. I think with all of these traits, especially with something like cannabis or something like schizophrenia, the environmental risk factors outweigh the input of your genetics. So even if you have a high or a low polygenic, whatever your polygenic risk score, using cannabis increases your odds of getting psychosis much more than your polygenic risk score, even having a very high one. But as that graph showed, there is some evidence that there's an interaction. So if you have a very high polygenic risk score and you use cannabis heavily, you're at greater odds of developing psychosis than someone who uses the same amount of cannabis, but less, if that answers your question.
You can talk to Isabelle in London, because I think Tony has come all the way from UCL to give his talk, and we're taking all his time. Thank you very much, Isabelle, and thank you for all your questions.